about whether testing drugs on animals is ethical, one of the main argument proponents use is that it has helped alleviate the suffering of people through the medical advances made in testing labs. This is not the case at all. Not only is animal experimentation morally wrong, but many of its failures have negatively affected people. One example of failure in animal testing is with the drug thalidomide that happened in the 1960s. According to the organization Animal Friends Croatia, scientists had created thalidomide to help stop morning sickness for pregnant women. Even though it was tested on many species of animals before it could be purchased, it caused an estimated 10,000 birth defects and thousands of fetal deaths worldwide. Many of the babies suffered from phocomelia, a disease that does not allow their limbs to develop correctly. Next, I have just a short list of drugs that were passed by animal testing but failed in the human species. In 2014, over 1 million animals were tested on in the U.S. alone. That's just including dogs, cats, primates, guinea pigs, hamsters, rabbits, and farm animals. This does not include the millions of mice and rats tested on each year because the government does not consider mice and rats real animals, and therefore experiments don't keep track of how many they test. Next, I'm going to discuss a few types of the testing done on animals. The first one is the Draize test. John H. Draize developed the Draize eye test to assess eye irritation caused by various chemicals. In this test, a substance is placed in the eye using the other eye serving as a control. The rabbits are restrained and their eyes are evaluated after an hour, and then after 24 hours, and sometimes up to 14 days. Some continue to be evaluated up to 3 weeks later. The level of irritation to the eyes is scored numerically by observation of the 3 major tissues of the eye. Rabbits suffer from redness, bleeding, ulcers, and even blindness, and are likely killed upon completion of the experiment. The next type is acute toxicity. Acute toxicity testing is used to determine the danger of exposure to a chemical by mouth, skin, or inhalation. For decades, acute toxicity testing meant poisoning large numbers of animals in lethal dose 50 tests, which are conducted until at least 1/2 of the test animals die. Other tests include acute tox class method and up and down procedure, which typically involve the use of a smaller number of animals. However, during these tests, animals will often endure excruciating pain. The next type of test is skin corrosion and irritation. Skin corrosion tests assess the potential of a substance to cause irreversible damage to the skin. Skin irritation tests determine the level of damage caused by skin, such as itching, swelling, and inflammation. Both tests are typically performed on rabbits. The skin irritation test is often referred to as the Draize skin test and involves placing a chemical on a shaved patch of skin and using another shaved patch as a control. The next type is carcinogenicity. A carcinogen is a substance or mixture of substances that induces cancer or increases the rates of cancer. The test chemical is administered orally, placed on the skin, or inhaled in a 2-year duration. Animal health is monitored throughout the study, but most information is obtained after the animals are killed and their tissues and organs are examined for evidence of cancer. Rodents are more prone to cancer than humans, making them poor models for studying carcinogenicity. The next type is neurotoxicity. Neurotoxicity tests aim to find out if a substance causes alterations to the nervous system. In the rat neurotoxicity test, the animals are given daily doses of the test substances for 28 days, 90 days, or 1 year. The rats are observed for physical and behavioral changes during the test and are killed at the end of the test period and examined for signs of neurotoxicity. Next, I'm going to discuss alternatives to testing on animals. The first alternative is cell cultures. Almost every type of human and animal cell can be grown in the laboratory. Scientists have managed to coax cells to grow into 3D structures, such as miniature human organs, which can provide a more realistic way to test therapies. Human cells have been used to create innovative little devices called organs on chips. These can be used instead of animals to study biological and disease processes as well as drug metabolism. Devices are already been produced and accurately mimic the lung, heart, kidney, and gut. The ultimate goal is to use these chips to create a whole human on a chip. Cell cultures have essential to key developments in areas such as cancers, sepsis, kidney disease, and AIDS, and are routinely used in chemical safety testing, vaccine production, and drug development. The next alternative is human tissues. Both healthy and diseased tissues donated from human volunteers can provide a more relevant way of studying human biology and diseases than animal testing. Human tissue can be donated from, for example, skin and eye models from reconstituted human skin and other tissues have been developed and are used to replace the cruel rabbit irritation test. Companies such as EpiSkin, MabTech, and Cell Systems now produce these tests in easy-to-use kits for companies to use to test their cosmetics and other substances. Human tissues can be used also from a person who has died. Post-mortem brain tissue has provided important leads to understanding brain regeneration and the effects of multiple sclerosis and Parkinson's. The last alternative is computer models. With the growing and sophistication of computers, the ability to model or replicate aspects of the human body is ever more possible. Computer models of the heart, lungs, kidney, skin, digestive, and musculoskeletal system already exist. These can be used to conduct virtual experiments based on existing information and mathematical data. And that is why we should end animal testing.